So thank you everybody for joining us today. I'm Tanya. I am from the ADRES team. Welcome to our joint webinar hosted by ADRES and Paul on the topic of Tangential Flow Filtration Technologies, Answers and Solutions for Practical Challenges Faced by Biopharma Users. The agenda for today is, Alessandra, can you please change for the next slide. So our agenda for today is Or from the ADRES team will present in a few words ADRES, and then Alessandra will present Paul, and then we will have a 45 to 50 minutes lecture about TFF overview, and then we will save 20 to 30 minutes for Q&A. I just want you to please note that we can't see you. This is a webinar format, Zoom format. So all the panelists, we can see ourselves and we can't see you, the attendees. So you can please write us questions during the session in the Q&A section below, and we will approach in the end. And if we will have time, we can also, I hope that we will have time to do live questions. This will be, you will need to click the raising hand button, and I will see that you have a question, and then I will open the microphone for you. So have a good time and good luck. And enjoy this webinar. Oh, stage is yours.
Thank you.
A minute, I will put myself on mute. Okay.
So hello to everybody. My name is Or Degany. I'm CMC Director at ADRES, Advanced Regulatory Services. We're very excited to host you today in this webinar, and we're thankful for our colleagues at Paul. Just giving you a glance around who we are. So ADRES is a biotech consultancy company, and we provide the full encompass of support for startups, early phase startups, and early clinical development phase, and also late clinical development phase for both pharma companies and biopharma companies. We provide the support in the worlds of quality assurance, CMC, regulatory affairs and scientific affairs, medical QA, and project management. And we can help you both as consultants and both as provide you become part of your employees for a limited amount of time according to the needs of the company, depending on which clinical phase you are. We are supporting a lot of companies in Israel and companies outside of Israel, in Europe and in the US. We could help you with due diligence activities. We have an SME status in Europe, which could really assist non-European companies in approaching the European agencies. And we also provide different kinds of training and courses that are tailor-made, especially for your needs, according to whatever you need. So that's about us, and I will leave the stage now to Marco from Paul, please.
Yes, thank you. So hello everyone and welcome. Thank you for joining this call on behalf of the Paul Corporation team on the call. This is Marco Sanvito. I'm part of Pal Corporation, more precisely of the Pall-Biotech business unit, working in the EMEA regional marketing team. So we would like to share with you a couple of insights about our corporation And first of all, would like to mention that Pall Corporation is today part of Danaher Corporation. Danaher Corporation is a US-based company. And as you can see on the slide, under the umbrella of Danaher Corporation, we have quite a number of different operating companies, as we call them. So you probably recognize quite almost all of these brands that you see on the screen. But more importantly, what I would like to mention is that thank you to this large family of companies, we have the ability to support end user throughout the entire life cycle of a pharmaceutical drug. So as you can see at different stages in this, let me call it journey, starting from lead generation and optimization through cell line development, process development, preclinical and clinical, till the manufacturing stage, the different companies under Danaher umbrella have the ability to support end users as appropriate. Please, Alessandra, next slide. Okay. And more precisely, we are working very close with Cytiva. You are probably familiar with Cytiva. Cytiva is the former GE biopharma division, and we are now part of the same Danaher family. And you see here an example of this capability that we have to support end user through the entire process. So here you see a number of what we call unit operation, each one covering a specific process step in a biotech or bioprocessing workflow, starting from upstream through clarification, chromatography, viral inactivation, and so on and so forth, till the bulk drug substance filling and the drug substance formulation and filling. We have this ability thanks to this combined portfolio of technology. And the last thing I would like to mention on this capability is that we have some standard configurable bioprocess platforms. You see here the most relevant ones, so monoclonal antibody, AAV, LV, and some customized one for specific process. And last but not least, mRNA manufacturing. So we can offer to end user standard but configurable bioprocess platform to cover a end-to-end solution. And with this, I will hand over to Alessandra to take you through the presentation for today.
I'm Alessandra, and I'm part of the European scientific and laboratory team, which help the customer understand and go through the trials processing and also all the know-how sharing between Paul and the customers. So I'm part of the European team, especially regarding the purification team. So we take over our upstream and downstream part related coming from the depth filtration to TFF and so on. So this presentation is meant to give you a brief overview of tangential flow filtration modalities and best practices, and to respond to frequently asked questions, which we will be glad to also answer after this presentation, so in our Q&A session. So this presentation will be divided in 5 sections, in which we will be focusing on TFF setup, key process parameter, some process-related questions, the scale-up processing, and finally the cleaning and normalized water permeability steps. But first thing first, how do we choose the right TFF membrane for our process? Firstly, we need to determine for what application the membrane is needed. So based on the target size of our molecule, the two main filtration methods will be the microfiltration or the ultrafiltration, which have different applications in bioprocess. Secondly, it should also be determined the right product variety available on the market. So the TFF membrane can come in many different configurations. The most used are cassettes, but there are also the holofibers, which are mainly used for microfiltration approach and also for viscosity, highly viscous product. And finally, we have the ceramic membrane, which are used for high viscosity product and also for their high chemical and temperature resistance. For this presentation, though, we will mirror to cassettes as part of our portfolio. So after that, we should also consider which should be the correct membrane cutoff. So based on our molecule size, we should, as a rule of thumb, we usually define the membrane to be 3 to 5 times smaller than our target molecule. And lastly, we should also consider different variables which come into play when choosing the right TFF membrane. Yes, the molecular weight cutoff play a key role in the choice, but it's not the only parameter to consider. We also have to consider the shape of our molecule, the charge, the conductivity, and the concentration. For this reason, usually a preliminary screening test might be suggested to determine the right membrane and cutoff to choose. So let's speak about setting up. Before start the processing of our TFF system, we should also consider that a number of steps must be completed before the actual process. Starting from the installation to the flushing and sanitization, we have a bunch of steps which need to be taken before starting our process. All these steps, together with the post-conditioning step, which include either the flush, the cleaning place, the normalized water permeability, should be considered when deciding how long our process should take. So we should estimate the time required for all these steps, including our processing itself. So be sure to consider all these steps when taking into account how long our process should take. How can we shorten this process? Well, the first thing we can do is for sure optimize not only our processing, so our concentration on diafiltration, for example, but also to optimize our flushing and cleaning steps. There's something that actually that can be done. And also this can actually help us to shorten our processing time. But also working with single-use TFF systems can shorten this processing time because, for example, but also every cassettes, we can skip some preconditioning steps. All conditions because the cassettes will be after the use will be discharged. So that's the two main way how we can shorten our process. So next test we will talk about the integrity testing. So why to conduct an integrity test? Well, firstly, to fulfill our validation requirement. So that's something that nowadays it's becoming clear that something that's need to be done. But most importantly, the integrity test can help avoid product loss and also time loss. because by detecting membrane or systems defect before starting the process, we can actually fix any damage or misplacement without losing our product. So the two main tests to be performed are the IT test, which tests if the membrane itself is integral, if there are no holes or scratches on our surface. Then the system which allow to detect any damage to valves and connection and to ensure that the overall system is assembled correctly. There are two main techniques to testing: the air diffusion, which is the direct flow measurement, and the pressure roll, which is an indirect measurement. During the air diffusion test, a pressure is applied to the system and the gas molecule diffuse through the membrane. The gas flow at a given test pressure is measured on downstream or upstream side, and it's compared with the maximum flows, which usually is given by the filter suppliers. The pressure decay test is an indirect measurement of a gas flow through a wet membrane. So when the test pressure is set up and after the stabilization, the gas inlet is closed and the pressure decreases during our testing period. The acceptable pressure decays is again usually set by the supplier.
Alessandra, are you? We can't hear you. Good. Wow. Maybe she needs to stop the video. It will help. And try to write her. Oh, she can't hear us. Just a minute, everyone. Sorry. And this is one acquisition it's a process which is supporting the filtration process. Hi everyone, sorry, I've just muted Alessandra because she can't hear us and we can't hear her good. So I wrote her and I hope she will see it. And we will be back in 2 minutes. Oh, let's see if we can hear her. Alessandra?
Can you hear me?
Can you speak again, please?
Can you hear me?
Yes. Okay. So please go on without the video. Sorry. Okay. I'm sorry for maybe its internet connection is always failing when needed. I'm so sorry about that. So I just. Okay. I will just wrap up the video because I think you could see. So I was speaking about the resistance phenomenon. The resistance of the membrane, it's usually a sum of the difference resistance which come into play. So we have the polarization resistance and the fouling, which is responsible to the decrease of process flux over time. So the resistance phenomenon, the polarization is the accumulation of molecules
so also described as gel layer formation, and it's an essential process in ultrafiltration. It can be optimized, and it could help to stabilize a steady-state equilibrium between the gel formation, the membrane permeability, and the back diffusion into the flow stream. While the fouling, it's a non-suitable process condition, so we don't really want to have it. It's the result of a molecule which can be trapped on the surface of our membrane, in our pore openings, or within our membrane networks. This happens because the membrane actually gets dirty, and the absorbed material works as an additional resistance to the permeate flow. The fouling phenomenon can mainly occur in microfiltration process, while the polarization phenomenon is typical of the ultrafiltration process. Although these two are key parameters and should always be taken into account. So let's have a look at the typical ultrafiltration optimization. With the protein solution at a constant cross-flow condition, the permeate flow will be proportional at low TMP, and then a plateau appears. So we do have an increasing of our permeate flow and transmembrane pressure until a plateau occurs. At this plateau, we have a limiting flow. So this can be explained by the concentration polarization model. The permeate flux increases linearly with the TMP within the membrane control region. While we go over this region, we have the polarization controlled region. And here, when we reach the plateau, there is an accumulation of protein on the surface of the membrane, as we saw before. So here, the permeate flux is now controlled by the concentration polarization. The system should be operated in this region, so the membrane controlled region. So in order to control the process, the TMP value should be stable. This is a key indicator. So if our TMP remains stable and we can control this process, we have a controlled and optimized process. When our TMP fluctuates, we have an uncontrolled process. Just to remind, the TMP is the transmembrane pressure and is the force that drives fluid through the membrane, carrying along the permeate molecule. And also another key and important things about our key operating parameters coming to diafiltration method. We do know that we have two methods when diafilter our solution, and we have the batch or stepwise method and the constant volume, so the continuous one. For the batch diafiltration dilution, the sample is diluted with an equal volume of water. This for sure reduces the concentration enough, as we can see also in this picture. When we concentrate to original to obtain the original volume, so we have actually half of the liquid that contain the salts, which are the blue dots, but the number of our protein concentration represented by the red dot, it's the same. So we had half of our salt concentration, but the same protein concentration. So we have actually reduced our salt concentration. When we repeat those dilution more and more, we can actually reduce the overall salt concentration. And when we arrive, for example, through the second diafiltration and the fifth diafiltration, if we dilute it 5 times, we can actually reduce the percent of the salts removed. So we can keep lowering that salt concentration. After 5 diafiltration, for example, we will have 95% of the original salt removed. But actually, this is a process which take a bit longer time and also a lot of buffer to be executed. While we speak about continuous diafiltration, we can actually use the buffer tank, which is connected to our feed tank, and usually we also have a pump which delivers the buffer at the same rate, so at the same speed as the permit rate. So this maintains a constant level in the tank, and which is really easy to do and also to scale up. The filtration volume can be calculated for salt reduction and also easily scaling up. So this table is the exemplification of what I was saying before. So the comparison between continuous and batch diafiltration, deciding that we have, for example, 100 permeable salt, it's more efficient to use continuous diafiltration than discontinuous diafiltration. So we can see it by the number of dia volumes. We have a significant difference between the two of them. When we have, for example, we arrive at 5 diafiltration, we have a 99.3% of salt removal with continuous diafiltration, while a 96.9 for discontinuous diafiltration. So the goal of diafiltration is actually desalting our solution. So our diafiltration should be stopped also when the conductivity of the permeate is below the target. The use of continuous diafiltration also help us save buffer, and also is less stressful for biological products because the process time is shorter and also the conductivity change is smoother. This is also really easy to automate the process and to scaling them up. Let's come into some process-related question. So this is one of the questions we hear the most. What about shear-sensitive molecule? So when working with shear-sensitive molecule, usually the straightforward approach, especially if we have an existing system, so existing TFF system, would be to adjust the process parameter. We can lowering the cross-flow flux and reduce the mass transfer process. Although this surely would be time-consuming because to re-optimize the process is actually time-consuming. And also we also need to consider that by lowering this value, we will enhance the number of pump passages, which actually is the main stressor for shear-sensitive molecules. So we need to pay really much attention to this kind of process adjustments. Another approach could be to change the technology used. So, for example, the holofibers can be used for gentler processes condition, but also here we do have we can have experienced the same issue as before. Another approach would be to choose a different technology, such as the single pass TFF. This technology, for example, our pulse cadence SPTFF modular kit, it's a technology that avoid the recirculation loop. So without recirculation loop, it becomes an optimal solution for shear-sensitive molecule. This technology, in fact, is a process development tool that provides flexibility, and also its use for challenging application. And it's, as I was saying, a single pass TFF. So our configuration allow to achieve higher concentration factor and also using less volume, and to work in continuous processing without having circulation loop, and also without having shear damage to our molecule. This also reducing working volumes and it results in high product recovery. So yeah, here we can see a comparison between the conventional TFF and our single pass TFF. We can clearly see that the concentration profile in conventional TFF occurs over time, while the cadence single pass TFF occurs over space. So as I was saying, we have completely different technologies that allows to have a shift in how we consider and how we process the TFF processing. And it's really helpful for this kind of application. When considering the bioprocesses stage in which the TFF is usually applied, the TFF process doesn't really require to be sterile. Therefore, power reusable cassettes are not claimed sterile, but they can be sanitized with appropriate agents, such as sodium hydroxide or sodium chloride and peracetic acid. The cadence single-use system cassettes are gamma irradiated, so they're actually sterile and ready to use and can eventually be operated in sterility by users. While, for example, Cytiva ready-to-process holofibers are especially meant for aseptic processing. So there's the main difference between this product and this definition. So coming to the scale-up processing, when we think about the scaling up processing, we should also consider a bunch of different variables which come into play. And this variable can be either product related, such as our product molecular weight, our contaminant molecular weight, the initial and final volume, our desired process time, feed and buffer composition, and temperature. But also we should consider our process-related information, such as our membrane choice, our product retention profile, average flux, and operating condition. So all these variables come into play when deciding whether is the scale up, the correct scale up. To scale up, usually we have a linear scale up. The volume of product is used in the test is divided by the test area, and it should be more or less equal to the process volume divided by the process area. This basically means that if the plan is to make an industrial process in 5 hours, the data should also be produced in 5 hours, not 30 minutes, using applying the same filter stress, which I know is not always the case and is not always possible, but that's something we should take into account when scaling up our process. In the graph, you can see a typical TFF process chart showing the different steps. So there are many options when performing diafiltration and concentration during the TFF process, but this configuration allows to diafiltrate with less buffer volume because we first concentrate, and then to don't stress too much our biomolecule with a possible toxic contaminant concentration, because we divided this step into 3. So this is one of the most effective configuration speaking about concentration in the filtration, but it's not the only one available or applicable. So the first step in determining the filtration surface is to optimize each of the steps that are within our process. So in our scale down trial, of course, for each step, the average filtered flux should be determined. So the filtered flux is calculated dividing the permeate volume by the filter test area and the time spent in each step. You can see here all the calculations are the same for each step. After knowing the average permeate fluxes for each step, the volume of permeate that we need to be produced in each step, and also the total process time, a simple calculation can be done to calculate the surface area that's required. So just remember the thing that we were saying before. So if we determine the average permeate flow in our laboratory trials, and we do not take more than 30 minutes, and then we plan to have a production time of 5 hours, maybe to have a less membrane area, we have to consider that maybe we could have possibly overestimated the average permeate flow. That's why usually in the scale-up processing, it should be considered a pilot plant, a pilot scale testing. So to just adjust this kind of operation and of measurements. So the cleaning procedure is meant to maintain efficient productivity by removing falling material from the membrane. At every other aspect of our TFF processing, also the cleaning procedure can be and should be optimized to ensure reliable, repeatable, and efficient, and also economical cleaning. The cleaning parameter typically controlled by the user are the type of agents used, its concentration, the temperature and time, and also water quality and cleaning cost flow. These parameters are process and product specific. That's why it became important to select the correct cleaning agents, because it should be chemical compatible with the membrane and also effective in removing the specific contaminant. For example, the sodium hydroxide is one of the most used cleaning agents for biomolecules. So how do the temperature and the time influence the cleaning efficiency? Well, we need to consider that the chemical reaction rate doubles with every 10 Celsius degree in increasing in temperature. Thus, the temperature of the cleaning solution should be as high as possible, obviously considering our system and module limitation. Also, by enhancing the contact time between the cleaning agents and the fouling material, the cleaning efficiency can be enhanced. The cleaning solution should also be pumped through the system under turbulent flow and generally with a Reynolds number between 3,000 and 5,000, which typically equates to a linear velocity of 1.5 to 2 meters per second, and with the transmembrane pressure as low as possible. We need to remember that we are trying to clean the contaminants on the surface of our membrane, so we don't want to push through the membrane our contaminants even more. So the cleaning can be also done for really dirty membrane in 2 steps. So performing the first cleaning and then removing our cleaning agents and put new cleaning agents to try to make an in-depth cleaning of our membrane. That's, as I already said, that something should be optimized, and it's really product and process specific. Also, the quality of the water play a key role and should take into account. So the quality of the water should be a minimum soft water, ideally deionized water or filtered at 0.2 micrometer. On specially concern are levels of iron, manganese, aluminium, and silicon in water. So a general rule, the water quality used for cleaning and preparing cleaning solution should be equivalent to one used in the process. So the normalized water permeability is an essential parameter for measure prior to use and either post-use. It's the measure of the ability of water to pass through the membrane, and it serves as a benchmark for the cleanness of the membrane. And that's why it's measured before and after the membrane usage. Normalized water permeability, also called NWP, it's calculated by dividing the permeate flux rate, usually expressed in liter per minute per hour per meter square, by the TMP and multiplied by the temperature correction factor. The membrane recovery should be also measured after each cleaning to check the effectiveness of the cleaning itself. The membrane recovery is a calculation to determine the efficiency of the cleaning, and it compares the normalized water permeability before the usage and after the cleaning step. So the acceptable recovery criteria must be defined by the user, but most of the time it should be over 80% of the initial NWP. When the system is reused, it's important to record all NWP measurement and plot against the number of cycles used and also to keep track of the trend of these values. The NWPs does not take into account the permeate flux of the product. We need to remember that we are speaking about water. So the membrane's hydraulic resistance, it's one part of the total hydraulic resistance, but it's not that the one that made the overall gel layer generated by the product. So therefore, a low NWP does not equally mean to a significantly lower permeate flux in terms of product process. So that's something we need to take into account when evaluating whether or not we can clean or either reuse our membrane after and not great NWP measurement. So here is an example. The main goal of our NWP measurement is to ensure that reusability of our membrane and also the effectiveness of the cleaning. It's usually plotted against a graph and with the initial permeability considered as a reference. So now we can see in blue, there is the NWP initial and then the NWP at the end, so after cleaning. And these two are really similar to one to another. So this is an ideal visualization of our NWP. It's ideal actually. So consider it as it is. The efficiency of the cleaning is shown by comparing the before and the after, as I was saying. So just remember the 80%, but also to keep track of this measurement along the time of reusage of our membrane. Keeping track of the different variables during the lifetime of a membrane has a crucial role in determining its lifetime. For the official information about this topic, the regulatory documentation to refer to is the technical report number 15 from Parental Drug Association. The membrane lifetime determination, it's made by a continuing monitoring of process performances and by measuring and tracking process parameter trends in time. This should be cohesive along the monitoring, and no significantly downward performance trend should be registered during the membrane usage. Above the parameters tracking consideration, also a reasonable change-out period should be considered. For example, in conjunction with annual shutdowns. The overall validation of membrane reuse is to demonstrate the process reproducibility, the absence of process measure carryover, and that the bioburden and the endotoxin level are under control and within the acceptable limitation. So now I will hand over again to Or to have a TFF must-have checklist.
Hello, everyone. I will share with you one slide regarding my experience, my personal experience from working in this field and from visiting other facilities. What are the basic must-have that you need, or kind of checklist for TFF when you approach, when you are approaching a commercial scale, or you already approached the commercial scale. So doesn't matter where you are in your life cycle, in their clinical development, any one of these lines here could be very useful to you. So when you are doing your development studies and you're trying to, and you're doing your attempts to develop your process, use the QBD strategy and try to define if you do have a critical process parameters in your ultrafiltration or diafiltration processes. This is a key issue in your process control and will be extremely important when you approach process validation to know if you have or not have critical process parameters in your TFF, in your UF or DF process units. So this is something very important and essential as you as part of your evolution of the process. You need to control and monitor your process parameters. This is extremely important. You have to monitor pressure, pH, TMPs, the permeate flux, and any other parameter that in general you would monitor for a TFF process unit, and those that are more critical to your specific product have to be even more carefully monitored. And please remember, one of the most important issues is all around control. Control is extremely important. Another thing is that this is an expectation when you go into the commercial scale, that each time that you approach your TFF unit back, after you haven't used it for 1 month, 2 months, 3 months, or 4 months, and the system has been stored with a preservative like NaOH, be sure that you wash out the preservative with your WFI. You check the last water rinse, and you confirm by checking the last water rinse that you have very low conductivity, which is a sign that you were actually able to remove out your sodium hydroxide or preservative, and do a test for bioburden or endotoxin and a microbial test. This is very important to show that you did not have a microbial accumulation in the period that the system was not used. Use relevant IPCs where required. So you might decide not only to have in-process controls after you finish the concentration, but maybe if you have a diafiltration and only after that the concentration, you might decide to have an IPC at the end of the diafiltration, and again have IPCs in place that are rational and provide you knowledge about the process as it goes on, and have a rationale why you are checking what you are checking. Okay, and this again relates to control and monitor your process. As Alessandra also talked before, you need to have a net water permeability procedure in place, which is indicative as an evidence of your cleaning efficiency. This is something very important to follow and have it as part of your routine procedures and operations in the commercial scale. Have a cleaning method in place. This is very, very important. And I will give you, let's say, the ideal situation is that before you approach process validation, you already did your cleaning validation, for example, on 3 engineering runs that you did before your process validation campaign. And in that way, you are already going into process validation with, you have a lot of confidence, you have a validated cleaning method, and you know that you're in a good place. However, companies, due to constraints in timelines and resources, sometimes go into process validation, and at the same time that they're doing the process validation, they're doing the cleaning validation. That is a higher risk. But in case you do that, and you, for example, after your first process validation, you tested for cleaning the equipment, and you failed in one of your equipments in the specification, please make sure to clean that equipment thoroughly until it meets its specifications, and only then go into your second process validation run. Remember, if you want to release these process validation batches, you have to provide evidence that the equipment that was used was cleaned before you loaded that material on that equipment. Please remember that. And as I said before, have in place a study to confirm lack of bioburden accumulation during storage period of your cassettes or holofibers. This is something that regulatory authorities and auditors ask a lot about. They want to make sure that if you didn't use your holofiber or cassette system for 6 or 8 or 9 months, and now you decided to do a campaign after 9 months, then they want to know that the cassettes or holofibers did not accumulate a large amount of bioburden, and now this bioburden is flushing now, going straight into your product. And the last issue that I want to talk today is about the lifetime study. So lifetime study, which is actually providing some evidence if you could use your system 20 times, 30 times, 40 times, or 50 times. So you should establish your lifetime study in a small scale model that is representative of your commercial scale. That's a very important study. You have to do it in an appropriate manner. And when you are ready, you did your process validation and you're ready for a BLA submission, have already a protocol that for a lifetime study that is a commitment that you intend to continue do your lifetime studies in the commercial scale. And the reason for this is also for extending your lifetime. So for example, if your lifetime study showed that you can use your cassettes 30 times, there is a huge effect of cost of goods on resins and TFF if you could use them more. And in commercial scale studies, sometimes we see resins being used 100 or 120 times. And you would also want to use as much as possible the TFF. And again, this has to be according to this commercial scale lifetime study. The criterion for your lifetime study, either in the development scale or in the commercial scale, you would need to set a criterion. It could be, for example, yield related. So for example, after 20 or 25 batches, you see a huge loss of protein due to gel layer accumulation. That could be one reason for defining the end of your lifetime. Another could be failure to clean your cassettes or any other parameter that you find that in your specific case could define the end of your lifetime of your cassettes. So that was my part, and thank you very much for listening to this slide.
Okay. So thank you, Or and Alessandra, for sharing your knowledge. Now we've got to the Q&A part. So I'll ask Or and Alessandra also to open the Q&A section. And we have already 3 questions. So please approach. I'll put myself on unmute so you can talk. Sandra, maybe you can open the video now if you want to try to speak with video. And please, the attendees, if you want to ask live questions, you can raise your hand by clicking on the raising hand button, and I will see it and I can open for you a live question. So, Or do you want to approach, or Alessandra?
Actually, I also want to present.
So please, I think maybe for the first 2 or 3 questions, I think they're more related to you, or I could also answer something.
Yeah, sure. Actually, I just want to present to you also Beryl Karinowski, which is also our colleague and is the subject matter expert. So maybe you also could, I would just leave the floor to him as he has over a decade of experience in TFF, and maybe it would be also really helpful for us in answering your question. So Gregor, if you want to take over the floor is yours.
Okay. Thanks, Alessandra. Hello, everybody. My name is Gertman. I'm part of the field support team at Pall for the European technology purification team, and I'm the subject matter expert for TFF. Going straight to questions, I already answered one question. That was the small scale for single pass TFF. So don't know if everybody looked at the chat. So yes, we do have small scale modules for single pass TFF. The smallest ones have 650 square centimeters in the shortest 4 in series configuration. This allows to test the single-pass TFF technology with approximately 1 liter of feed material to characterize product properties. And if you like to have some really quantitative test for scale-up, typically we use like 2 to 3 liters minimum, or up to the maximum load of the membrane. And a true continuous process that could easily exceed the 40 liters if you run the single-pass TFF for several days. Single-pass TFF is available in different flavors. So we have the inline concentrator, which is a standalone module that Alessandra has shown. It only needs the connection of 4 hoses, feed, retentate, and the 2 permeate outlets to run the module with a pressurized source or a feed pump. Or you can go to the more controllable technology, which is the standard single-pass TFF modules that would require a retentate control by, say, an automated control valve at scale. This technology is then available with the 4 to 9 in series different path lengths and enables you for full control and precise adjustment, whereas the inline concentrator, as the product name implies, is an inline concentration tool only. Going for the second question on cleaning on single-pass TFF. So cleaning on single-pass TFF, the generic protocol foresees cleaning in true single-pass, but obviously this requires a lot of cleaning agents. So to reduce the amount of cleaning agent required, there is an option to run the cleaning for single-pass TFF with some recirculation. We would typically approach it by having a initial cleaning in single pass to wash out all the impurities and the dirt. And then for the remainder part of the cleaning protocol, it could be a recirculated approach. As any other TFF cleaning, this needs to be validated and qualified. Would refer similar to the PDA Technical Report 15 for validation of TFF for this. And if I go to the next open question, pore geometry considerations for TFF membranes. TFF membranes are qualified by the vendor with a vendor-specific test. So there is a nominal molecular weight cutoff assigned to the membrane, but there might be subtle differences between membranes of different vendors. So typically vendors issue their qualification, which kind of testing is performed to classify the membranes, and that gets you an idea. A 30 kilodalton and a 30 kilodalton membrane, like for retention of antibody, is not really differentiating. The main properties that are different would be the chemistry of the membrane, whether it's a PS membrane or regenerate cellulose membrane. That could have a different impact with regards to membrane fouling or the compatibility to the product. Imagine dealing with solvents in ADC processes, for example, where we would recommend a regenerate cellulose membrane. Obviously, it is recommended to test it and to see if the membrane is retentive or the impurity clearance is sufficient. And you can also, for partially retained membrane, check if the polarization of the membrane surface, that means the TMP applied and the cross flow applied, has an impact. But it's not like that we would have a universal characterization across the board. So there might be subtle differences evaluating certain membranes, whether these are, say, like our Omega membrane, a thin skin layer membrane with a very fine sharp cutoff on our PS membranes, or whether it's a more traditional membrane approach like our regenerate cellulose membrane, which is more like a solid membrane structure. The next one up on technical questions, which permeability rejection coefficient should one usually expect for buffer components, e.g. tris? So a buffer component is so small, any of those salts, that they should be 100% permeable and should easily pass through the membrane. What can happen is if you have some interaction of your retained species of your protein that potentially carries some charge, that there could be some interaction, and you would then see a co-concentration of the respective counter ion with the retained charged species. This is called Donan effect, And typically this is addressed by modifying the buffer composition of either the feed material or the diafiltration buffer, so that this co-concentration of the counter-ion species is addressed and you will still attain the target concentration in your final buffer. Scroll down. For single pass TFF, when starting with 1 liter, what will be the final volume? In a single pass TFF setup, we were able to get down to working volumes around 30 to 40 milliliters for the feed side inlet to the retentate outlet. But that means that there would be literally no material left in any of those vessels that we use to feed the material. So realistically, like a 100 to 200 milliliter final volume, so with 1 liter, maybe a 5 to 10x concentration is possible. If you are aiming for a higher concentration, obviously you need to start with a higher starting volume. Is there a
Gregor, we have one question from Dima Shamarakov. I will allow him to talk, so it's okay.
Yes, please.
Let's try it.
Do you hear me, Tanya?
Hi,
Yes.
Dima.
Hi. My name is Dima Shamrakov.
You can ask your question. I
From
can't hear you.
Okay. I confess I'm quite a rookie in the field of tangential flow filtration. I'm trying to desalt and concentrate quite a concentrated suspension of liposomes, lipid suspensions. It has a distribution from microns, from single microns to about 30-40 microns. And I'm trying to rid of submicron particles. And of course, I have a severe problem of a membrane fouling. I use a microfiltration with 0.1 to 0.2 micron holes. My question if it is doable at all, and how could I optimize this process if it is feasible at all?
Thank you. That's a very difficult question you're asking. Fractionation is typically working well if they are significantly apart in size, either molecular weight or size. If you are working with a membrane where the cutoff of the membrane is in range of the species, say that you have like the whole particle distribution from submicron up to 40 micron at a relatively even particle presence, then it will be very difficult because there will be particles that are blocking your membrane. If you have a like large 40 micron species and then only submicron particles, that could potentially work better. If you don't have this, the only option is to check if the permeate flow is too high, and that would cause a premature membrane fouling. So what you can do, you can check for the stability of your process at increasing permeate flows. So the critical flux theory. So you would start with the permeate flow closed, and then you would incrementally check for operating conditions with a fixed permeate flow, like 5 LMH, 10 LMH, 15 LMH. And while performing this in total recirculation, monitor the pressures. If you are not experiencing a pressure increase, you can go up 1 increment until the point where you would see within the first 2-3 minutes an immediate pressure increase. That is where your process is no longer stable. And then you would run at the sustainable flux, which is like 60 to 80% of this critical flux when you surpass the critical flux. It's called the critical flux theory. It might help, but for myself, I'm not 100% sure. This is typically applied with a clarification of bacterial feed or other substances. But my cells, as they are soft, I do not have sufficient experience if this will work, but it would be the suggested approach. Shall I go back to answer?
Sorry. Yes, we'll take one or two questions and then we will finish the session. I'll just write in the chat. We have two emails that all attendees can approach and ask questions, and we will be happy to help. So Gregor, please, the stage is yours for 2 or 1 questions.
I'll go from the down question. What type of Paul filter do you recommend for very viscous solution? That's also a very good question. Depending on what product we refer to and what state, we can process to very high concentrations with very viscous fluid, like for monoclonal antibodies, up to 250 grams per liter with single pass TFF technology. And the product at this final high concentration is highly viscous. That would be an option. Say we would go for the final UFDF, concentrate to the intermediate concentration, run the diafiltration on TFF skid, and then we would use like from 50 grams to 250 grams a single pass TFF to get to this very high final concentration. If you have a viscous solution from the very start, then the only way to get around is to use a lower resistance device. That could be either a suspended screen TFF cassette, a hollow fiber with a large lumen, or in the most extreme cases, some ceramic elements that are, say, likely typically used in the dairy industry, but also in the biopharma industry. You would use those for purification of antibiotics with filamentous fungi fermentation, for example. And then if we have time for...
We have time for 1 more, I think, and then thank you.
1 more. What is the most crucial for sensitive molecules? Maintaining stable TMP or a tentate flow? For sensitive molecules, it's definitely the flow. So the flow is not only causing a shear and a pressure drop in the device itself, but in the entire loop and also in the pump head. So specifically the pump head that agitates the whole recirculation is actually the main source of shear. So for shear sensitive products, I would always recommend to go with the lower cross flow. And it's also very unlikely that they are that impacted by the TMP in the first place. The CFF is definitely a primary parameter to be controlled. And with that, practical database is the last one. Unfortunately, I don't really have a solid answer to this. There is nothing, not really a one place to go. There are, say, options depending on where you are. Scientific literature is available, like refer to the publications of Andrew Sidney, for example. But unfortunately, there's no such online database at one place.
Okay, Gregor, thank you very much. Thank you all, PAL team, Alessandra, Gregor, Eran, Marco, and thank you, Or. Thank you everyone for joining us today, and I will see you in our next webinar. Thank you all, bye-bye.
Thank you.